Good morning. I want to welcome you all to our panel on AAV success studies looking at retinal degeneration and spinal muscular atrophy. I'm Dr. Joan Miller. I'm a retina specialist, and I'm Chief of Ophthalmology at Mass Eye and Ear and Mass General, and Ophthalmologist-in-Chief at the Brigham and Women's. We have a great panel today, and I want to introduce our panelists. First, we have Ken Mills, who's the CEO of REGENXBIO, Eric Pierce, who I know well, who is MD, PhD, and pediatric ophthalmologist and inherited retinal disease specialist, as well as the director of the Ocular Genomics Institute at Mass Eye and Ear and a professor of ophthalmology. Ron Phillip, who's the COO of Spark Therapeutics, and Meredith Schultz, who is a pediatric neurologist and executive medical director at Novartis Gene Therapies in Translational Medicine. So before we move into our discussion, I want to share my thoughts on how gene therapy has really changed our profession and our approach to patients and their families. I'm a retina specialist by training, as I said, and when I finished my training in the 1990s, there were really no treatments for inherited retinal disorders other than observation, counseling, low vision aids. Now with the success of Lexterna and the many clinical trials underway for patients with other blinding conditions, there's really great excitement among patients and their families, and the best and the brightest in our field are attracted to this area of ophthalmology. Zolgensma, of course, was developed for spinal muscular atrophy, or SMA, a lethal disease, not a blinding one, that affects our youngest patients. A family friend has a child with SMA, and I have some inkling of the fear of families and the promise of a life-saving cure. We have a great panel today, as I've mentioned, and we're going to discuss some of the lessons learned from the development of these two products and what we can hope for in the future. So thank you, panelists, and thank you, audience. We're looking forward to questions also coming from the audience as we go through this. My first question, I think this will take a little time from all of our panelists, is to really think about the lessons that we've learned from these first two first-in-class approvals. And Meredith, I would like to start with you, because you were our principal investigator in several of the pivotal gene therapy trials for spinal muscular atrophy. And you're now at Novartis working to bring gene therapy treatments to other indications. So what are the lessons learned? And if you could also reflect a little bit on the role of the academic center, the timing of transition to industry, and the role of pharma. I'll probably cut you off if you go too long.
Sure. Yeah, no, I certainly could talk about this for quite some time. So, yeah, it was quite an exciting journey for for the SMA community and for Novartis Gene Therapies, and for my experience as well. So, I had the opportunity to treat a number of patients with gene therapy, with Zolgensma, as a PI on some of these pivotal studies, and it was really the life-changing effects that this therapy had that really inspired me to join industry. And my work at Novartis Gene Therapies now is to drive the early development of our clinical programs beyond spinal muscular atrophy into other diseases. And from Zolgensma, we certainly have learned quite a lot. I think from a translational medicine aspect, and for many other aspects, we've really learned that there's not going to be a one-size-fits-all approach for gene therapy. So each indication, each patient group, each disease is going to have its own nuances, and really requires extensive collaboration between many of the different teams, between research, clinical development, manufacturing, others. So, it's quite a hefty task. And really, as we have learned many of these lessons from Zolgensma, what we're able to do is to take those lessons and really build a platform that will be able to set us up for success in the future. Really, I think we are just at the tip of the iceberg with what we have learned from gene therapy, and really, just at the beginning of being able to unlock the promise of this technology. So there's still so much to learn, and we're excited to be on that journey.
Thanks so much. Eric, I'll turn to you. You obviously were involved early in the development of Lexterna, and now are involved in many gene therapy trials. And if you could sort of address some of the lessons learned, and also, how did we end up treating a retinal disease as the first indication for an inherited disease?
Sure. Right. That's always been an interesting question. Why do we have the first approved drug for an inherited disease as a gene therapy drug as a retinal disease? I think that goes back in part to the history of gene therapy. The death of subjects in clinical trials, like the ornithine transcarbamylase trial, the Penn in the 1990s, and 6 others in trials of gene therapies for coronary artery disease, really, I think, made the field stop and think about additional vectors that provide additional safety, and then highlighted the advantage of local vector administration. And I think those forces kind of led to a focus on retinal gene therapy. And with AAV demonstrated to be effective at transducing retinal cells back in 1997, that really catalyzed work on inherited retinal disorders. That was followed by preclinical demonstration of efficacy of RPE65 gene therapy in a large animal model with observable improvements in vision in 2001. And that really supported moving to a clinical trials, plural, which started in 2007, and ultimately the approval of Lexterna in 2017. And in terms of lessons learned, we're really hoping we can replicate that Lexterna success for many other genetic forms of inherited retinal degeneration. As Meredith said, each of these disorders has unique aspects, but I think the general platform or paradigm of gene augmentation therapy can really be broadly applied. And I hope we've learned how to do that and can now effectively translate those therapies to many other disorders. And we've also learned that measurement of visual response to these therapies is not always straightforward. Sometimes we need novel endpoints, such as the visual navigation course, or MAZE, that was developed for the RP65 gene therapy trials. And I guess I'll stop there.
Right. Well, thanks. I may move on a little bit. And Ken, I was just interested, we've learned lessons from these first 2 indications, but are we ready now to go into broader indications? And, first, maybe some similar indications. I know REGENXBIO is exploring some of those. And what are some of the challenges, and, are we ready to go?
Yeah, Joan, thanks. I really like what Eric said about, one of the steps and lessons learned here was how to use the technology that we had in hand and, focus its delivery to tissues and cell types and disease areas that could be transformed with the gene therapy tools that were in place. REGENX formed at a time when that exploration was ongoing, but also brought new tools to bear that allowed us to reach into additional tissue types, additional routes of administration efficiently and safely. I definitely believe that, one of the lessons learned from the 2 approvals is we can and should continue to deliver AAV treatments safe and effectively. And, how we do that and how we navigate through that is definitely a function of the technology and the science that's available to us. When I think about the effect that Lexterna and Zolgensma have had on the field, it's incredibly profound medically. Just really unbelievable results. I think things that we couldn't have expected from other forms of modalities. How to address additional diseases, though, I think there was a little bit of a misconception that the immediacy of applying the Lexterna science or the Zolgensma science was going to result in more treatments. And in fact, we needed additional science to get into some of those adjacent tissue types, like instead of the RPE, getting into photoreceptors, or finding other ways to get broader distribution throughout the CNS, maybe not just focused on lower motor neurons in SMA. And that's where the expansion of trials has gone on, the use of new technology, and a lot of the things that I think Meredith was referring to we're focused on now. I believe we are there, Joan. We just started a late-stage clinical development program in wet age-related macular degeneration focused on expressing a therapeutic antibody, a protein out of cells, not changing the intracellular function to address a disease, to address an underlying pathology. And that's a major leap from the first-order approaches of thinking about intracellular monogenic gene replacement. So I believe that the foundation has been laid with some really excellent medical success and frankly, commercial success with respect to Zolgensma as well. Where we're going next, though, is definitely a function of science, continuing to be focused on the right diseases with the plan that companies are putting in place. And then I'm excited. I see this field like recruiting people like Meredith, Ron, in addition to people like Eric who've been in the field. We're getting people in place now to develop these great new treatments in a way that, honestly wasn't there 10 years ago.
Oh, great. And Ron, also, you obviously have, I'm sure, some thoughts on the lessons learned from Lexterna and how you're thinking about broadening to other retinal disorders and other indications would be great to hear.
Yeah, no, obviously our experience is a little bit unique because we were the first to launch an in vivo gene therapy in the US. And so a lot of the things that came up during that process, it was almost like an enormous task in trying to identify which areas to really focus on because there was so much that you could really spend your time on. And I think looking back at the experience, throughout the value chain from research to development to the manufacturing process as well as the commercialization, really the things that really stand out for us is probably mostly on the commercialization side. And, they're probably broken down into like 4 major areas. First one was really gene therapy market development is incremental to disease area development. I think most of us on the commercial side have been very used to launching new products. So you spend a lot of time in getting the marketplace aware of what the disease area specific information is. I think for us with Lexterna, it was that plus the gene therapy market development. There's a lot of misconceptions and myths around what gene therapy is. It goes from designer babies all the way to stem cells to even within the actual gene therapy field. Is it cell therapy? Is it gene therapy? What kind of gene therapy is it? Is it in vivo? Is it ex vivo? So that, lentiviral versus AAV. So there was just a tremendous amount of time that we needed to spend just educating both the physicians as well as, and through the physicians, the patients on what we were trying to do, at least with Lexterna and what in vivo gene therapy was all about. And the second piece is on the physician side, you're really moving away from the chronic treatment kind of paradigm to this one-time infusion, and that changes the care continuum for them. And so getting them comfortable with what the actual procedure is, as well as the after the procedure care of their patients was an important step. The second lesson learned is really around, I think around payers. Payers were, especially when we were launching, were very apprehensive about what gene therapy would mean for them on the budgetary side, and really demystifying what Lexterna was, what gene therapy is at this point in time was an important step. And then, really going as early as possible to them to actually listen to them and hear their issues and what their concerns were and what they wanted to see in the solution was an additional kind of important step in the process. Third one is really around coordination from manufacturing all the way to administration. We ended up needing to use specialized treatment centers to infuse Lexterna, and so getting the seamless integration from the product being made just in time delivery to actually working with the actual treatment center staff, the pharmacy staff, the receiving docs, to get the actual product to the operating theater in the time frame that's needed, and making sure that every step of the way is thought through was pretty important, especially early in the process. So getting that done and doing an individual kind of focus on those treatment centers is extremely important. And then the last one I would say is, and this probably goes across for all of the gene therapy players, including the cell therapy. I mean, these are disruptive therapies and they require model disruption. We really need to teach both internal in our own companies as well as external stakeholders to forget what they know. I think all we know is the chronic world, chronic therapy treatment world. We really need to get people to think about what's possible with these one-time treatments and what are the new solutions, and whether it's outcomes agreements or varieties of ways to test kind of the efficacy of the product into multiple years. There's a lot of creativity that can be brought to bear, and I think we're just starting to scratch the surface on that. So there's a lot more innovation that's needed, not only in the technology, but in the delivery of the technology.
Thanks, Ron. Let's move a little bit to a discussion on safety. I mean, these two are very successful, and does this mean that we're now just unleashed to move forward? Is AAV such a safe therapy we don't need to worry about it, or are there still some safety issues that we need to consider? Meredith, I'll start with you if that's okay.
Yeah, you bet. So this is certainly a very important discussion, and it's really critical that we maintain the trust and the credibility with the patient communities, throughout this process. We are constantly monitoring for safety signals in our Zolgensma program, not only, as part of the clinical trials, but as part of the long-term follow-up of those patients, and also through real world evidence. It's a challenge because this is a one-time therapy. Patients don't always want to come back. There is somewhat of a notion that patients are cured after they get treated, which has been really a challenge to make sure that everyone is educated to ensure that these patients come back, get adequate follow-up, continue to see their care teams, and are monitored for safety signals. We are really trying to innovate and look at creative ways to be able to monitor these patients long-term through home visits, remote monitoring. So any method that we can use to make it easier for the patient to stay connected. And ultimately, as we learn as a gene therapy community, continuing to share those findings and, make every clinical trial a little bit better, a little bit safer, and to make sure that we're really sharing all of those learnings together. So, forums like this is great where we can, make sure that we're all passing on, important information with safety and efficacy and other advances in the technology.
Great, thank you. Ken, we've had some safety issues sort of early in the history of gene therapy, and then done quite well. But there have been some recent burps, I would say, particularly in the ophthalmology realm. I just wondered what you think on about safety, especially as we go into these less life-threatening or blinding situations.
Yeah, I mean, Eric certainly alluded to the history, and I do think by the mid 2000s or since around 2010, generally clinical trials and commercial launches have been safe and well tolerated, and it's been a real expanded universe of different disease and tissue targets. Certainly, we've seen some AAVs require treatment for drug-related, typically transient clinical symptoms. As the gene therapy itself is reaching sort of pharmacological steady state. And I think often this trade-off, like you're alluding to, Joan, is easily absorbed against the background of some of the benefit risk that we see in some of those diseases. But, and for the most part, I mean, a lot of the diseases in clinical study for gene therapy are very sick patients with advanced disease. So as we move into new areas, in addition to continuing to explore that, I mean, I really like what Meredith said about the uniqueness of sort of the one-time element here. I mean, in the clinical trial setting, we have the advantage of, in general, patients, coming back for follow-up. And, that changes entirely in the commercial setting. And when we have these one-time treatments that are delivering the type of outcomes that we're seeing with things like Lexterna and Zolgensma, it does create a new set of challenges to get, I think, ourselves as companies comfortable, to get stakeholders like physicians comfortable about how do we approach follow-up here? I often talk to my team about, parallels to transplant, except there's not even the sort of chronic immune suppression requirements post-transplant where, patients and families need to stay in contact with the clinical center. This is very unique. I think that, and we have to have important and creative ways, new ways to monitor. For us as a company, like we're very careful about going into new areas of research, clinical research, in areas where benefit risk is different than high infant mortality rate examples. And in those cases, we look at the foundational safety profiles in other, disease studies that have been done. We look for the absolute cleanest safety profiles, and we prioritize those routes of administration and those approaches for AAV until we get comfortable that some of these newer routes of administration and newer technologies have similar foundational work. So, I think we're well on the path of understanding a lot of the safety of AAV in different tissue types and with different routes of administration. But I do appreciate what Meredith is saying, that the monitoring on the other side of approval and the learning that comes out of that, and we're just a couple of years into these first approvals, is a real high rich content area for growing and understanding. And it should feed back into both preclinical and early clinical development.
Great, thank you. Eric, I know you know that I'm really keen on neuroprotection. I'm just wondering, when we're going to be able to apply gene therapy to that kind of a strategy. And, is there science, we can talk about the science of neuroprotection, but is there other science in terms of AAV and gene therapy that we need to really hone before we broaden out that much?
Yeah, first of all, I think we will expand to those kinds of indications like neuroprotection, so it's coming. I think we'll be able to tackle those applications as we get more experience, as we were just discussing. But I do think, as Ken, actually as all of us have started to say earlier, this is a place where more science is needed. We need to continue to develop vectors that allow for additional delivery approaches and higher efficiencies of transduction of the specific target cells that we're really going for, because that allows us to reduce dose and reduce toxicity. That's going to be really important as we get to more common therapies like neuroprotection or more common disorders like age-related macular degeneration, as we were discussing. In the retinal disease field, this has been trying to find a vector, for example, that transduces effectively following intravitreal injection instead of subretinal injection is kind of a holy grail. I'm hoping we're going to get there, but we're not quite there yet. But as you suggested, I do think there's great potential for AAV vectors to deliver other cargo, right? Like neuroprotective agents instead of therapeutic transgenes, but also the toolkit needed for gene editing or base editing or prime editing or optogenetics. And I think those are all coming as the science matures and as we understand how to reduce adverse events with improved vectors.
Great, thanks very much, Ron. We may be closing in on our final few minutes, but I just wondered if you had an addition to add on that in terms of going into common diseases.
I think just to add on, I think manufacturing capacity, things like pre-existing neutralizing antibodies, managing the immune response, better targeting, a lot of the things that Eric said, we really need more innovation in order to go after some of these, more advanced kind of monogenic as well as polygenic diseases. And slowly, I think it's happening, right? I think there's enough gene therapy companies. This is kind of a network effort. There's enough gene therapy companies working on a variety of these things, especially in the in vivo portion. There's also kind of technology leaps that are happening even on the non-viral side. So, it's an exciting time. Things are being looked at. We still have to have a little bit of patience to let that play out. And I think over time, these solutions will start to come through. The future is bright, but it's almost expected. I mean, we're still working with almost first generation technology, right? And so there's that next generation that needs to come forward in order to go after some of the more complex diseases that are out there.
Great, thanks. Meredith, maybe in our last minute, I know there are issues about sort of scaling up and the manufacturing, and I'd be interested in your thoughts on how much of a challenge that remains.
Yeah, as everybody here has mentioned in the panel, the manufacturing space is continuing to evolve, and I do think we're going to continue to get more efficient over time. Right now, we don't have the manufacturing ability to treat, very common diseases, but I think we will get there. There's some unique ways that we are thinking about approaching this, which is taking a broad indication and really looking at a small part of that population that have similar clinical or disease physiologic characteristics, with the hope that eventually we can translate that into the larger population. So, I do think that we'll get there, as we've all mentioned, this technology, will hopefully get more refined over time so that we can, tune it up, tune it down, turn it on, turn it off, as we need to over time, and that it sounds like science fiction, but, we're not that far off, but
great.
certainly, lots more work in the field is needed.
Thanks so much, and I want to really thank all of our panelists. It was wonderful just listening to you and great questions and answers by you. We're going to take a short break, but before you leave, please take a moment to answer a couple of polling questions, which will appear on your screen, and then want everyone to reappear at 10:55. Thanks so much.